Welcome back, guys. So today I will discuss on the life cycle of Puccinia, graminis tritici. So this Puccinia graminis tritici, if we take a look at its systematic position according to Ainsworth, 1973, it belongs to the kingdom Mycota, division Eumycota, subdivision Basidiomycota, class Teleomycetes, order Uredinials, family Pucciniaceae, and genus Puccinia. So this fungi, it is a plant pathogen, a very common plant pathogen, and it occurs about 1800 species. Okay, 1800 species, they are distributed throughout the world. 1800 species of Puccinia. Now, out of these 147 species, they are reported from India. And these 147 species, they are known to cause serious diseases of cereals that such as wheat, oat, maize, and barley. Now, the species of Puccinia, they are obligate parasites. Okay, we know that obligate parasites are those parasitic organisms that require a suitable host to complete its life cycle. So, without exploiting a suitable host, these obligate parasites, they cannot complete their life cycle. They cannot reproduce, they will fail to reproduce if they cannot obtain a host. So species of Puccinia as well, they are known to be obligate parasites. And these species of Puccinia, they may be autoecious and heteroecious. Okay, autoecious meaning they need only a single host, only one host to complete their life cycle. Whereas heteroecious, they need two different types of hosts to complete their life cycle. So many species of Puccinia, they may be autoecious or they may be heteroecious. So the wheat crop in India that gets damaged by the different rust diseases caused by these different species of Puccinia, the most common one, they are black stem rust, okay, black rust, which is causes by which is caused by the Puccinia graminis tritici. The brown rust on the leaf, which is caused by Puccinia recondita. The Puccinia striformis, which causes a yellow rust. Okay, so these are the most common rust disease caused by species of Puccinia in wheat crop in India. So in this particular video, we are going to discuss on the life cycle of Puccinia graminis tritici. We know that Puccinia graminis, it causes stem rust of different cereals like wheat, rye, oat, and many other cereals. And one more thing is that it is a host specific. What it means that the isolate which we obtain from, suppose from wheat, it cannot infect oat or it cannot infect rye, and vice versa. The isolate which is being isolated from oat, it cannot infect wheat. This shows that this is a specific pathogen. Okay, it indicates their host specificity, meaning Puccinia graminis is very host specific. And the wheat crops of the northern and the southern region of India, they are largely affected by this pathogen, Puccinia graminis tritici. So we are going to discuss to take a look on its life history. Now, Looking at the life history of Puccinia graminis tritici, first thing we must know that this Puccinia graminis tritici is a heterogeneous. Okay, it is a heterogeneous pathogen. Why is it called a heterogeneous pathogen? Because it requires two related hosts to complete its life cycle. Now, the two hosts required by this Puccinia graminis tritici, the host which it infects during its life cycle, is Triticum aestivum, that is wheat, and Berberis vulgaris, that is a barberry plant. So this is regarded as the primary host, and the Berberis vulgaris is the alternate host. Now, the Puccinia is also macrocyclic. Okay, why is it called macrocyclic? Because it has an extended life cycle. We will study that throughout its life cycle, it can change into different spores. Different spores are germinating during its life cycle. Okay, during the different stages of its life cycle, and these spores are uridospores, telutospores, basidospores, pycnospores, and the achiospores. Also, because it can produce many forms, many spores, that is five types of spores in its life cycle. That's why it is also known as polymorphic, because poly means many and morph meaning form or structure. So it produces five different types of spores in its life cycle. That's why it is known as polymorphic. Now, if you take a look at the vegetative body of this fungi, we know that the vegetative body of fungi is mycelium. Okay, so the mycelium is the vegetative part of a fungus consisting of a network of fine white filaments. So this is just an example in this picture to show it is looking like somewhat like this. Okay, these fine, very fine filaments. And actually fungi, they have different colors of, we can see different colors of the fungal mycelium. So the vegetative body is it consists of a network of fine white filaments known as the hyphae. This is the microscopic slide. Inside a microscope, we can see a network of these fine lines of hyphae. Okay, so a network of hyphae is known as a mycelium, which is a vegetative body. The single fine filaments here, they are known as the hyphae. So a mycelium is a network of hyphae. Now, the hyphae, if you enlarge, this is the enlarged picture of a single hyphae. Now, the life of this Puccinia, in the life cycle of Puccinia, it actually can form two types of hypha. The two types of hyphae, that is monokaryotic and dikaryotic. Now, what do we mean by monokaryotic mycelium or hypha? Is that we can see that in monokaryotic, it is characterized by the presence of only a single nuclei in a cell. Okay, you can see in these cells we can observe only one nuclei, whereas in dikaryotic mycelium, you can see it is characterized by the presence of two nuclei. So these two types of mycelia, they are produced by the by Puccinia. They are formed during the life cycle of Puccinia. Now, the dikaryotic mycelium, it occurs in wheat plant, that is the primary host, whereas the monokaryotic mycelium, it usually occurs on barberry plant, that is the alternate host of the pathogen. So next, these mycelium, they are actually they are very much branched. Okay, these are just an example to show that the mycelium of the Puccinia, they are very much branched. They show branching. and these mycelia, both the dikaryotic and monokaryotic, they have septation. Okay, you can see here. For example, in this case, you can see a septum. So the mycelia of Puccinia is also septate. Even in this microscopic slide, we can see septation. Okay, so just remember this, that the Puccinia also, it shows septation. It has septation and much branched. Okay, and also in near the septum you can find the pore. Okay, the pore through which the uh, germ tube will exit upon germination. So the pore also are present. The septum also is present in the mycelium in both the monokaryotic as well as the dikaryotic uh, fungi, dikaryotic mycelium of this fungi. Now, these um, this figure, it shows that the Puccinia species, they grow intercellularly. Okay, you can see here they grow in the spaces between the cells. That means they grow intercellularly in the intercellular spaces, and they have a hostoria. You can see these are the hostoria, which helps the fungus in the attachment, as well as the as well as to penetrate through the host organism, and to absorb nutrition. Okay, that is the function of the hostoria. You can see here the spore when it germinates. This is the germ tube, the apressorium, which peers down through the host epidermis and grow intercellularly with the formation of the hostoria. This is another example which shows the spore first. When it germinates, it will form a germ tube, the apressorium. This is the penetration peck which will start to penetrate downward, and in this case, this is the intracellular mycelium. I just want to show this figure in order to make you understand the difference between intercellular and intracellular. In intracellular, they are growing within the cell as well. But in intercellular, they are growing in the spaces between the cells, in between the cells, in the intercellular spaces, with the formation of the hostoria, which help in the which help in the penetration of the host cell. Now, the fungal cell wall, they are composed of chitin, glucans, and also the membrane proteins. Okay, the membrane proteins, and also the cytoplasm of this Puccinia. Just remember this, that it also contain vacuoles, oil globules, and the glycogen bodies. So now let us study the different types of spores which form during the life cycle of Puccinia. Now the different types of spores, they are this Puccinia, it produces 5 different types of spores. We have already seen that is the uridospore and telutospore, which grows on wheat. That is the primary host. The basidiospore, which we will study later in detail, that they grow on the soil or on dead plants upon soil when they are still attached to the dead plants. Okay, and the pycnospores and aciospores, which will form on the barberry plant, that is the alternate host. So let us take a look at the first type of spore, that is the uridospore. Now, this uridospore, because the life cycle, it is a cycle, it is a cyclic process, remember, macrocyclic. So here, let us start from the uridospore. The uridospore, actually, they are formed from the aciospore. We will study this in detail also about the aciospore, but just remember this, that the uridospore, they are formed from the aciospore. Now, the uridospores, they are born on Uridosaurus. Okay, let me just show you this picture. You can see here the symptoms of Uridosaurus um, on the leaf of a wheat plant. So here the uridospore, they are born on Uridosaurus. Now the Uridosaurus, the these Uridosaurus, they are formed on the plant, on the wheat plant, that is the primary host. Okay, and you can see here the symptoms. They are brownish in color, brownish or red in color. Now the Uridosaurus, they are the spore producing body. Okay, so they will produce uridospore. So here the uridospore, they are born on these Uridosaurus. Now the Uridosaurus, they develops on wheat plant from a dikaryotic mycelium. Remember that we have seen that both the monokaryotic and dikaryotic mycelium is present in the life cycle of Puccinia. So in this case, on the wheat plant, the Uridosaurus, we have the dikaryotic mycelium. Now the dikaryotic mycelium, it is produced by the germination of these archaeospores. So clear that these archaeospore, they will come in contact with the host epidermis, that is the wheat plant, and they will start to germinate. So upon germination, they will form a dikaryotic mycelium. Okay, so these archaeospore, we will see later that they comes from the barberry plant, generally through wind dissemination. Now the archaeospore, after reaching the wheat plant, they may attack the leaf, the stem, or the legumes. And on germination, the archaeospore, they will produce a dikaryotic mycelium. So after they produce a dikaryotic mycelium, this will enter through the stomata. Just take a look at this figure D, that this is the host epidermis, the host cell, whereby this dikaryotic mycelium, the germinating archaeospore, will pierce down, will enter through the stomata. You can see here, these are the gut cell. Okay, and this is the vesicle. They are piercing down, they enter through the stomata and they grow intercellularly, forming the dikaryotic mycelium. Okay, these are the secondary dikaryotic mycelium which form in the intercellular spaces. Now the mycelium of the subepidermal region, they develop erect hyphae. Okay, the hyphae, they are erect, and these later on, they will produce a binucleate uridospore. Now remember, this is very important to remember that the spore which is produced here, which is produced due to the germination of the archaeospore, it is binucleate. It is a single cell, oval in shape and brownish in color, but it is binucleate. Now the uridospores, they will develop in groups. Okay, they usually develop in groups under the epidermis, called the uridosaurus. Now these uridosaurus, they appear in the form of reddish brown pustules, just like in this figure I have shown, the reddish brown pustules which appear. Outside, these are the uridosaurus which form due to the development of the uridospore under the epidermis. Now, with maturity, this host epidermal, it will rupture. Okay, it will rupture by pressure. It will burst due to pressure. Okay, the pressure due to the development of these spores. So this host epidermal wall, it will burst, and the uridospores, they become exposed. Okay, so if we take a look at a single uridospore here, it is oval. I had mentioned it is also unicellular, brown, thick wall. Okay, and it has these germ pore it has these germ pore from which the germ tube will exit, okay, upon germination. Now the spore wall, you can see the wall here is acinuleate. Okay, it has an acinuleate outer layer. Acinuleate, that is, it is set with small spines. You can see here the outer layer, it is set with these small spines. So we call it acinuleate outer layer. Now what happened here, this is the same figure which shows this apricerium, the guard cell, and how this apricerium will, how this germinating spore will pierce down into the lower epidermis, and it will start to form the secondary germ tube, the dikaryotic mycelium. So here what I said is that the uridospore, now in favorable condition, okay, in favorable condition, that is, the favorable condition for the uridospore is usually a winter season, okay, it will again germinate. Now does infect another healthy wheat plant and develop next crop of uridospore. Now the procedure, it may repeat several times in a single season. So several times it can repeat in a single season. So now, but what happened at the end of the season, at the end of the wheat season? Okay, so what will happen to the uridospores at the end of wheat season? Actually, these uridosori, which produces uridospore, they will start to produce another type of spore, that is the telutospore, along with uridospore. So at that time, this uridosori or the uridosaurus is known as a mixsaurus. Okay, it is not called as a uridosaurus anymore. At that time, it is known as a mixsaurus. Why? Because it produces, at the end of the wheat season, it starts to produce another type of spore along with these uridospore. They will produce another type of spore, that is the telutospore. But the mixsaurus, okay, the uridosaurus, which is now known as the mixsaurus, it will gradually convert solely into telutosaurus. Okay, so the spore producing uridosaurus, which produce a uridospore, now becomes a telutosaurus, which will produce telutospore. Okay, so at the end of the wheat season, now this uridosaurus, it is converted into telutosaurus, which will produce other type of spores, that is the telutospore. So here the telutosaurus, they develop exclusively by the infection of the uridospore. Okay, so in the wheat plant, due to the infection of the uridospore at the end of the season, the the wheat season, so these telutospores will start to develop. Now those red pustules, brownish red pustules, now they have become black. Okay, so you can see the telutosori, they look black raised streak, okay, that develop on the leaf sheath, and also on the stem. So that means at this time you will understand that the wheat now, it is infected by other type of spore. Actually, it is infected by the uridosori at first, but these uridosori at the end of the wheat season, they gets converted into telutosori, okay, which appears black, which look black raised streak. Okay, so here the telutospores, now if we take a look at the telutospores, they are stalk, they have a stalk, they are spindle shaped, thick and smooth walled. So unlike uridospore, you can see here, which has an acinuleate outer layer, the telutospore, they are actually smooth walled, okay, with round, okay, with with either round or pointed. You can see here it is slightly pointed apex, and also two cell, okay, two cell and slightly constricted. You can see here at this part where the septum, where the septation is present, it is slightly constricted and slightly constricted at the septum. Now the spore, they are, just let me show you this microscopic slide. The spore, they are chestnut brown in color, okay, and they are measure about 15 to 20 micrometer into 40 to 46 micrometer. Now each cell is a dikaryotic having one germ pore. So let me show you this diagram. Each delutospore, it is a dikaryotic. What is a dikaryotic? Characterized by the presence of two nuclei. So here not only two cells, but in each cell we will have two nuclei. So here also two nuclei, and this also two nuclei. Okay, so that condition is known as a dikaryotic. So each cell is dikaryotic, having one germ pore. In this figure you can see here this small germ pore here at the top of the apical cell. Now this is the apical cell, the upper one. The germ pore is present at the top, whereas it is present below the septum. Now this is a septation, so just below the septation you can find here another very small pore, that is the germ pore in the lower cell. So at the apical cell it is present on at the top, okay, at the apex here at the top of the apical cell, and just below the septum in the lower cell you can find another germ pore. Now I had mentioned that it is through these small pore in the outer wall of these fungi, of these fungal spore, that it is through these that the germ tube will exit upon germination. So we will study this one. Upon germination, the germ tube will emerge, or it will exit from the germ pore, from these germ pore. Now both the nuclei, okay, both these, because there are two nuclei in each cell, both these nuclei, they will fuse together, okay, and form a diploid nucleus. Now this is a diploid nucleus. So the delutospores also, they become exposed to the outside environment by rupturing the host epidermis, okay. Same, just like the uridospore, when they grow, they are dividing and more spores are producing inside the host epidermis. The host epidermis will burst, okay, it will get ruptured and releasing the spore to the outside environment. So same with the delutospore. Now the delutospore, they become exposed by rupturing the host epidermis. So these delutospores, they actually act as a resting spore. Okay, remember this, that they act as a resting spore. Now, why they called a resting spore? Because they can survive under unfavorable condition. So under unfavorable condition, these delutospores can still survive. So they are known as the resting spore. Now, as with the onset of favorable condition, okay, with the onset of favorable condition, now the favorable condition is that low temperature with high humidity, okay, for the delutospores to germinate. Now the favorable condition is low temperature and high humidity. So with this favorable condition, this delutospore, they will start to germinate on the soil, okay, either detached or attached condition with the dead host. Now I had mentioned earlier that we will see that the basidiospores, how they develop, okay, during the in attached condition or either detached. they will germinate on the soil and form other type of spores, that is the basidospores. This is another picture of the Luteal You can see here some are round and some have a pointed apex. So the basidospores, you can see they form from the telutospore. Now it develop on germination of the telutospore. So the telutospore, it germinates by producing one germ tube. You can see here this one and this one. So one germ tube from each cell. You can see here these are the telutospore and one germ tube from each cell is formed. Now this germ tube, it has a limited growth. Okay, it has a limited growth and is known as a promycelium or a probasidium or epibasidium. Okay, so what happened in this case after the formation of this germ tube? Now the diploid nucleus, remember that the nucleus, they fuse together to form a diploid nucleus. These nuclei, they have fused together to form a diploid nucleus. So the diploid nucleus will pass through this germ tube. Okay, it will pass inside the probasidium and it will undergo meiosis to form 4 haploid nuclei. So the 4 haploid nuclei, 2 of which are positive and 2 of negative type. They are of different types. Okay, it's just the type of haploid nuclei which form. They are different. They are different that from each other in the genetic material. That's why they are known as the positive type and the negative type, just to differentiate between these 2 types of spores. So that means nuclei. 4 haploid nuclei will form 2 of the positive type and 2 of the negative type. So here the probasidium now becomes 4-celled. 1, 2, 3, 4. It has become 4-celled, the probasidium, by a transverse wall. Okay, there is a transverse wall formation and it has become 4-celled. So each cell then will develop a small projection. You can see here from this figure C and this figure E. Now there is a small projection which will form. Okay, so the sterigma through which the nucleus and the cytoplasm will push outside and develop basidospores. So the nucleus as well as the cytoplasm from these cells, they will push out to this projection. Okay, so forming the basidospores. Okay, now these basidospores, remember 2 of them, they are of positive type and 2 are of negative